Hello everyone. Today we are going to see a topic production of baker's yeast. Introduction. Baker's yeast Saccharomyces cerevisiae is a single-celled fungus essential in baking and fermentation industries. It is responsible for leavening bread by producing carbon dioxide during fermentation. It is also used in various industries such as baking, brewing, and winemaking process. And it also acts as a dietary supplement due to its nutritional value. Microorganisms used for production. Species Saccharomyces cerevisiae. The characteristics of Saccharomyces cerevisiae are single-celled fungus and it converts sugar into carbon dioxide and alcohol by fermentation, and it is a fast growth microorganism. It is highly metabolic efficiency and it is easy to cultivate. Raw materials for production. Carbon source. Molasses, glucose and sucrose. Nitrogen source. Ammonium salts, urea or protein isolase. Minerals and vitamins such as phosphates, magnesium and biotin. Factors affecting production of baker's yeast. Temperature. Optimal range is 30 to 35 degrees Celsius. pH. Ideal range is 4.5 to 5.5. Aeration. Adequate oxygen supply is critical for yeast growth. Nutritional balance. Proper ratio of carbon, nitrogen and trace elements. Stages of yeast production. Preparation of culture medium, inoculation, fermentation, harvesting and drying. Microbial propagation process. First, starter culture. A small amount of yeast culture is introduced into the medium. Batch fermentation. Yeast cells multiply by dividing and consuming sugars in the medium. Then cell growth. As the yeast grows, it consumes sugars and produces byproducts such as carbon dioxide and ethanol. Industrial scale up. The yeast is then transferred to larger fermenter for mass production. By this cultivation process, the Saccharomyces cerevisiae is multiplied. Harvesting and separation, centrifugation, filtration, concentration, and then purify. In centrifugation process, it is used to separate the yeast cells from the fermentation broth. In filtration process, to remove residual solids or unfermented material. In concentration, yeast is concentrated into a slurry. Purity, ensuring the yeast is free of contaminant like bacteria or mold. Drying process. In drying process, there are three types of drying: spray drying, fluidized bed drying, and vacuum drying. In spray drying, converts the yeast slurry into dry powder. Fluidized bed drying ensures a more uniform drying process. Vacuum drying, a more energy efficient method and preserving yeast activity. Forms of yeast. After drying process, the yeast is converted into three forms: active dry yeast, compressed yeast, or liquid yeast. Then packaging. Yeast is packed in vacuum sealed containers to prevent moisture uptake. Quality control and standard. First, microbial testing. Ensure no harmful pathogens or contaminants are present. Then, purity test. Verifying the yeast strain and ensuring no genetic drift. Activity test. Measure the fermentation ability of Saccharomyces cerevisiae before packing. Shelf life. Ensuring yeast remains viable for the required time period. Challenges in yeast production. Contamination control, preventing bacterial or fungal infection. Environmental factors such as temperature fluctuation or pH imbalance can affect fermentation. Raw material quality. Consistency of raw material such as sugar and nitrogen source can affect yeast yield. Waste management. Disposal of spent yeast and other byproduct can be an environmental concern. Thank you for listening.